Bismillahirrahmanirrahim. I am very thankful to the organizing committee for giving me the chance to present in International Brain and Biomedicines Conference organized by Department of Physiology, Government College, University, Faisalabad. The topic that I am going to discuss today is production of gallic acid under solid state fermentation utilizing waste from food processing industries. My name is Dr. Shagufta Saeed and I am working as an assistant professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. Gallic acid is a phytochemical compound that is present in various parts of plant as secondary metabolite. It is colorless to slightly yellow color in and crystalline compound, melting point of 252 degrees centigrade. It has a wide range of applications in pharmaceutical industry as an antioxidant agent, antibacterial, anti-cancer, and precursor of antibacterial drug called as trimethoprim. In food industry, it is used as food preservative. Mainly, gallic acid is produced by hydrolyzing tannin using the acid hydrolysis or enzymatic hydrolysis. On the other hand, some microorganisms have the ability to hydrolyze tannin by producing the enzyme called as tannase. And tannins are naturally occurring water-soluble polyphenols that are present as secondary metabolites in various parts of plants such as wood, bark, leaves, peels, seeds, fruits, and galls. This is a list of various microorganisms that have the ability to produce tannase. Conventional production of gallic acid by chemical hydrolysis of tannic acid is unfavorable due to poor yield, high cost, and discharge of lethal effluent that cause environmental pollution. So, the present research was designed with the hypothesis that fruit waste that are rich in tannin have the potential to be used as substrate for the production of gallic acid by solid state fermentation. These were the objectives of the study to evaluate the potential of different fruit byproducts that are rich in tannic acid to produce gallic acid by solid state fermentation using Aspergillus niger, and then the optimization of different physiochemical parameters for the production process. Aspergillus niger was first obtained from the first fungal culture bank of Pakistan, University of the Punjab. It was then maintained on Sabarot, dextrose agar and preserved at 4 degrees centigrade for further use. For suspension was produced by adding 10 ml of 0.9% sodium chloride solution to the plant of fungus until it was further diluted till the spore suspension of 3 into 10 raised to power 7 spores per ml were obtained. It was used as inoculum for the fermentation studies. These were different substrates that were used like peels of banana, pomegranate, mango, apple, and the fruit seeds of jamun, black plum, apple, mango, and tamarind. Peas and seeds were dried and then ground to a fine powder. Then this powder was stored at room temperature to be used as tannic acid substrate for solid state fermentation. First of all, the tannin content of the substrates were estimated by spectrophotometric method. Then the substrates were screened for achieve the production of gallic acid. Different substrates were taken 5 grams of each substrate in different flasks, and the water ratio was set at 1:2, pH 4. Autoclaved, then 1 ml of inoculum was added and incubated at 30 degrees centigrade for 72 hours. And then the substrate that gave the highest yield of gallic acid then was further selected for the optimization of different conditions like substrate water ratio, incubation period, inoculum size, and pH, temperature, effect of addition of different carbon sources and nitrogen sources. After the fermentation was completed, then 50 ml of distilled water was added to the flask and kept at orbiter shaker at 150 rpm for 10 minutes, then filtered, and the gallic acid was estimated by mixing with methanolic redonin, and the absorbance was taken at 520 nanometer by using the spectrophotometer. Then the crystallization of gallic acid was done by Soxhlet method. Gallic acid produced was identified by FTIR and purity was estimated by HPLC using the standard of sigma Aldrich. All the experiments were carried out in triplicates and the data was analyzed by one-way ANOVA on SPSS software. These are the results of the study. This shows the growth of Aspergillus niger on saprots dextrose agar plates. Tannin content of different food byproducts, as it can be seen from the table, that German seeds had the highest amount of tannin content. Therefore, from different substrates selected, German seeds yield the highest amount of gallic acid by fermentation. Then further conditions were optimized using black plum as the substrate. A highest yield was obtained at 1:3 of substrate water ratio, incubation time of 96 hours, inoculum volume of 2, pH of the basal media set at 5, 30 degrees centigrade of incubation temperature. From different carbon sources, glucose gave the highest production. None of the nitrogen sources had the ability to give the highest yield of gallic acid. These are the crystals of gallic acid. FTIR spectrum of gallic acid. The red one shows this power sample, while the blue one is the standard, and it can be seen that similar peak pattern was observed. This is the HPLC chromatogram, and 99% yield, 99% purity of gallic acid was estimated. At the conclusion, different fruit phases were tested for the gallic acid production, from which the highest yield of 13.85 milligram per gram was obtained using the German seeds. These conditions can further be utilized on pilot scale to obtain highest yield of gallic acid for the commercial needs. These are the references of this study. Thanks for your attention.